Foundation of America. Welcome and thank you for attending tonight's webcast, CCFA Research Initiatives, Genetics and Microbiome in Inflammatory Bowel Disease. I would like to address a few housekeeping items before we begin. To allow full participation in today's program via the web, please be sure to disable any pop-up blockers on your browser and computer. After the presentation, we will open up the program for your questions. We will take as many questions as time allows from the webcast participants. If we are not able to take your questions, our IBD Help Center can be reached Monday to Friday, 9 a.m. to 5 p.m. Eastern Time by calling 888-694-8872 or via email at info@ccfa.org. Upon exiting today's program, you will be prompted to complete a brief program survey. We ask that you please take a few moments to provide your responses, as your feedback is extremely important to us for planning future educational activities. Finally, if you are experiencing any difficulties hearing the program, you can listen via telephone by calling the following toll-free number, 1-877-309-2071, access code 822-669-285. Within 1 month, you will be able to access the recording of this webcast on our website. For this and our other webcasts, please visit www.ccfa.org/resources/webcasts.html. I now have the pleasure of introducing our speaker for today's program, Dr. Thaddeus Stappenbeck. Dr. Stappenbeck received his MD and PhD from Northwestern University Medical School. He trained in anatomic pathology at Washington University Medical School and is board certified in this specialty. He studied host microbial interactions in the intestines as a postdoctoral fellow. He serves on the Scientific Advisory Council and as a co-chair of the Senior Research Award Study Section for the Crohn's and Colitis Foundation of America. Dr. Stappenbeck's research program has focused on understanding the pathogenesis of inflammatory bowel disease. We are very excited to have such a wonderful speaker here today. Without further ado, I will now turn the program over to Dr. Stappenbeck.
Thank you, Cassandra, for that nice introduction, and it's my pleasure to be able to speak with everyone this evening. So I guess we'll get into today's objectives for this conference, and so there are 3 main objectives. And the first goal is for me to communicate with you basically the role of genetics and the microbiome in inflammatory bowel diseases. So the Crohn's and Colitis Foundation is basically set up 2 initiatives around both of these kind of ideas, and I can give you some insight into where these are actually heading. Second would be to basically do exactly that, highlight recent advances in CCFA research initiatives within these 2 areas, and then third, to learn how that you can be involved with IBD research. Okay, so the next slide. So there's an audience poll.
The broadcast is now starting. All attendees are in listen-only mode.
So the audience poll question is we'd like you to basically respond to, is which basically describes you, and you can see the following options. You're the patient with Crohn's disease, patient with ulcerative colitis, family member, patients with Crohn's disease or ulcerative colitis, a healthcare provider, or no connection, you're just interested. Okay. So the next slide. Okay, so let's to get started, let's just talk about simply basically what are inflammatory bowel diseases. So these basically, they include two major subtypes called Crohn's disease and ulcerative colitis. These basically affect a growing number of Americans each year. The current number is 1.6 million Americans. And there are roughly 70,000 new cases that are diagnosed each year. So this is roughly 1 in every 200 people in the United States. So this is becoming an increasingly common disease within our society. This is also the incidence has been increasing over the last 100 years. Basically, the incidence of this disease has doubled every 20 years. So this is something that's been growing in magnitude over really the last century. For the disease, the disease course of disease, symptoms, and prognosis are very different from one person to another, and this I think is what has made this disease so challenging. This really appears to be a constellation of diseases potentially, and this is what I think has made kind of developing novel therapies extremely challenging. So the next slide. So the two major types are basically, as I mentioned, are Crohn's disease and ulcerative colitis. So the major effects of both of these diseases are in the intestine. Crohn's disease, so they have basically overlapping but distinct both pathologic and clinical manifestations. And so I think the best way to think of these is kind of two ends of the spectrum. Crohn's disease at one end of the spectrum and ulcerative colitis at the other end of the spectrum. Crohn's disease is characterized typically and classically by inflammation that occurs at the distal part of the small intestine. So the small intestine is basically the major absorptive organ within the body for basically various food particles. So basically, and Crohn's disease in this area is basically characterized by patchy, full-thickness inflammation. So what does that mean? That means that there's inflammation that basically occurs throughout the entire thickness of the wall. So this includes the lining epithelial cells, as well as the muscle that lies beneath this. So this becomes thickened and inflamed throughout. While this classically occurs at the distal small intestine, these lesions actually can occur anywhere in the gastrointestinal tract, all the way from your mouth all the way to the distal portion of the colon at the anal rectal junction. So that's again, this is something that's not really understood why this happens, but it has this very patchy nature, and because of the fact that there's this full thickness inflammation, it's not uncommon then to see things like fistulas or abscesses or strictures develop, because you're basically causing inflammation to the outer wall of the intestine, and this can lead to these complications. And these I think are very severe complications to this form of the disease. The disease does have extra intestinal manifestations that go beyond the gut. Some of these are shared in common with the other subtype, ulcerative colitis, and these can include inflammation of your biliary tract, of your eyes, and a variety of other places in your body. But the major focus, I think, here we'll be thinking about the inflammation that occurs in the intestine. Ulcerative colitis, on the other hand, is kind of at the other end of the spectrum. So instead of basically having kind of areas of patchy inflammation throughout the GI tract, this basically classically is more of a superficial type of inflammation of the inner lining of the gut. This affects primarily the intestinal epithelium and the cells that lie immediately beneath it. And this is, instead of being patchy, this tends to be continuous. So this is inflammation that typically begins at the anal rectal junction, and then basically kind of over time can march more proximally away from the anal rectal junction. And so you typically get a large area of ulceration that's basically continuous. And so classically, because of this, this is basically something that is typically just seen in the rectum or colon. So as I mentioned, that these are two ends of the spectrum. There are, and diagnosing these two particular entities can be quite challenging, and it requires basically understanding of clinical history, the endoscopy that the gastroenterologist does, and what the pathologist sees from biopsies taken by the gastroenterologist under the microscope. And this constellation of findings then can basically classify people as Crohn's or ulcerative colitis. Though there is a proportion of people that have what is called indeterminate colitis. That's a subpopulation. Okay, next slide. Okay, so the CCFA research goals basically are to basically try and identify groups of patients, new therapies, and ways to optimize effectiveness of current available treatment. So basically, what this comes down to basically is being able to, I think, classify and subclassify this disease more precisely, so that we can better determine which patients will better respond to specific types of therapy. So that's basically the overall goal. The CCFA is also really, so to actually do these studies, you need investigators to do this, and there are always new ideas coming into the field, and a lot of these new ideas come from new or from investigators who are kind of new to the IBD field. And the CCFA has been really wonderful over the years at basically creating these types of links with people who haven't thought about doing IBD research, and basically drawing them in and getting them interested in thinking about these problems. So this has been, I think, a major goal of CCFA, and I think it's been really successful over a long period of time. And third, the goal of CCFA research is basically to advance quality improvement in IBD patient care. So this is ultimately our goal, is basically to improve the lives of patients with IBD. So everything that we're doing in terms of the genetics and microbiome initiatives has this goal ultimately in mind. Okay, next slide. Okay, so how will research then into IBD, where can this actually take us, and what are some of the major big questions that are basically present? So so the first is trying to understand the root cause of these diseases. That we still don't really, we're gaining a better understanding of why they occur in general terms, but what we'd like to know is specifically in specific patients what actually triggers these diseases. These are diseases that occur to basically healthy people up to a certain point. You develop this disease, and then basically you can more or less have this for the rest of your life. So trying to understand how this actually occurs, and what role does genetic susceptibility play, and then what role do environmental factors such as the microbiome, what do they actually play in this disease? So this is really, I think, a big something that's really very much on people's mind doing research. The second is basically trying to then take this information and basically develop better medications that can induce remission and prevent relapses in all patients. So this may very well be that we'll be able to personalize therapy in the future based on subclassification of the disease. That's my current bias and thinking about this. But this is, I think something that we all want to be able to take the basic research we're doing in genetics, microbiome, and basically then translate this so that this is something that can be developed into new therapies. The third thing is basically preventing some of the severe complications that I mentioned, particularly for Crohn's disease, such as fistulas, obstructions, and abscesses. These are things I think that really are quite debilitating. And even if there were therapies that targeted specifically these particular types of complications, they would be extremely valuable, even if we don't yet know how to prevent the disease. And another complication that's mentioned is cancer. There is a small but significant risk of developing bowel cancer in patients that have IBD, and so trying to understand how that occurs and how to prevent that is actually very important, and is an important mission of the CCFA. And fourth, we basically would like to basically be able to predict who high-risk individuals are, maybe based on genetics, and then develop methods for actually preventing the onset of disease. So this I think would be a long-term goal, where we'd be able to take this information and begin to figure out who's really at risk and what kind of things can we do to prevent high-risk individuals from developing the disease. Okay, next slide. Okay, so the factors that we think about in terms of the cause of IBD are numerous and complex. And this is kind of a classic slide that's shown basically when basically you're trying to tell people what are some of the major factors to consider. So there are four basically major factors. I've mentioned some of these as we've gone along here. The first at the top are basically genetic factors. The second are environmental exposures. These can include everything from the foods you eat to particles in the air and water that are things that you would be exposed to in your gut. A third element of this that's often lumped within environmental exposures, because it's often linked to them, is the gut microbiota, which we'll talk a little bit more about. And the fourth thing is your immune system. So you have a very kind of large and diverse part of your immune system is present within the wall of your gut, and the process of inflammatory bowel disease basically creates a system of dysregulation, basically, where your immune system then becomes hyperactive and reacts to things, particularly the elements of the gut microbiota, where it basically spins out of control, and then its activity can't be reined in easily. So these are really, I think, four of the major factors that we are really trying to understand within this space. So we're going to focus really on two of these today that are highlighted now, the genetic factors and the gut microbiota. So next slide. Okay, so the first thing we're going to talk about is the Genetics Initiative. I believe there's going to be a short video.
Thank you, Dr. Stappenbeck. If you wouldn't mind muting your line for a moment.
Researchers around the world have solved a major mystery by identifying more than 100 places in the human chromosome that contain gene mutations now proven to contribute to Crohn's disease or ulcerative colitis. You can imagine treating inflammatory bowel diseases like a game of darts. The bullseye is the cause of the disease we're trying to treat, and the dart is the treatment. Until now, treating inflammatory bowel diseases sometimes felt like playing darts without a bullseye. Not really knowing the cause we're trying to change. Now we have several new bullseyes to go after. That's the breakthrough. That's tremendous progress. And to us, that's very exciting. Our job in the Crohn's and Colitis Foundation Genetics Initiative is to mine these amazing data. Our work will continue, developing new therapies, finding more pathways that lead us to better and better treatment approaches.
Back to you, Dr. Stappenbeck.
Okay, so that really I think that really nice video, the person narrating that video, I'll just mention, is a very close colleague of mine, Dr. Skip Virgin. He's also at Washington University, and I've had the pleasure of collaborating with him now over the last 7 years, specifically on this area of trying to understand genetics in inflammatory bowel disease. So he seems like a great guy in the video, and he is a great guy. He's a great scientist. So I just wanted to say a few shout-outs to him. Okay, so genetics. So what we're going to talk about here basically are so Skip mentioned that there are 163 gene loci that have been identified that lead to increased susceptibility for either ulcerative colitis or Crohn's disease. And the really interesting thing is, as geneticists have begun to map out these mutations, what has become, and they've now done this amazingly in now tens of thousands of patients with inflammatory bowel disease. What has actually emerged is basically what I mentioned when I was talking about the kind of the clinical manifestations of ulcerative colitis and Crohn's disease, that these are essentially a spectrum of diseases, and the genetics that underlie both ulcerative colitis and Crohn's disease also follow that spectrum as well. So there are a number of these 163 genes that are basically more Crohn's disease centric, and there are a number of these 163 genes that are more ulcerative colitis centric. And then, interestingly, there are a number of genetic loci that seem to be in common between these two diseases. So I think that's been pretty exciting, is that the genetic underpinnings of the disease seem to match what we see in terms of even the clinical manifestations of the disease, that it is heterogeneous. So the things that are now really exciting with now looking very in great detail now at these gene mutations that occur that lead to susceptibility to either UC or CD, we can now begin to try and understand what some of the functional changes that occur. So when you see these susceptibility mutations, what genes are they linked to? How do those genes function? And then how do those mutations then lead to susceptibility? So at some level, this is we take this gene by gene. But at some level, we have some higher level types of studies that we can do using something called systems biology, where we can then try and take, this is a large and complex set of genes, and then basically what we try and do is functionally annotate them, and then basically try and figure out, are there specific pathways that are then abnormal within either ulcerative colitis or Crohn's disease, or even these patients that are kind of lumped in the middle genetically. So this is basically the overall goal of the Genetics Initiative. So the next slide. So I have mentioned a lot of this in the background. So what we're really focusing on here are these 160 loci, and the focus of the Genetics Initiative is really to study the unique loci. So we're very interested in genes that basically are more skewed to Crohn's disease or ulcerative colitis. And the goal really, some of these genes we know something about, but many of these genes we really don't know much about. So basically the approach is where there are genes that we don't know much about them, is to basically try and do basic tests on cells and model organisms such as mice, where we can begin to understand how these genes actually function. What do they actually do within the intestine? And then how might mutating these genes then predispose to disease? So that's a huge part of this goal. For genes that we know a little bit more about their function, then we try and push this a little further into studying human tissues and cells to understand how they might be related to or involved in susceptibility to Crohn's disease. And the overall goal basically is to use these then, as Skip mentioned, we're looking for bullseyes, and these are potentially bullseyes. So we're trying to figure out what these specific targets are, so that we can then, if we find it, if we have a patient that has a particular mutation, we can hopefully design drugs that will basically be able to target that mutation in that specific gene. So that's really the big goal here, to be able to do this. Next slide. Yeah, and then this is basically just in cartoon form what I've just said, basically, and using Skip's idea of the bullseye, basically, is now is that we're able to take these specific genes. We have a way of actually prioritizing these genes within our group to study, and then basically looking for the best targets for new therapies. And we basically as things progress, if they remain interesting, we keep working on them and keep trying to develop them so that we can pass them on to pharma, so new drugs can be developed. And if they turn out to not be interesting, we basically put them aside and don't work on them anymore. Okay, next slide. Okay, so that's the genetics initiative. So now we'll talk about the microbiome initiative. So this is I think at a very I think exciting but very different type of approach than the genetics initiative. So the goal here is basically to try and understand how the microbiome might might play a role in either inciting the disease or play a role in basically perpetuating the disease. So so we've done a lot of characterization of the types of microbes that live within our intestines, and there have been really spectacular developments in the last 10 or so years, and basically for us to be able to go in and actually take surveys of microbes that actually are in the gut and define exactly who is there. And this has been, I think, very illuminating and very exciting at the same time. So, so the basic ideas here, what we basically found is that there may be bacteria that may be for lack of a better word, better to have in our gut that we would want to promote their existence within our intestines. And there appear to be other bacteria which, for lack of a better word, are bad, and basically are bacteria that we would like to discourage from being in our intestines, and these are associated with disease. Next slide. So, basically the overall goal here is to basically try and, much like the genetics initiative is looking for the role of specific genes that could trigger disease, this kind of takes the flip side of this, where we're interested in specific microbes that can actually play key roles in triggering inflammatory bowel disease. So the microbiota is normally a very think of it as a very complicated, basically, type of ecosystem. There are literally, if you look at the species level, there are literally 1,000 or so different species of bacteria that live within our intestine. So it's very complex. It's also a very large in number. There are in the order of 1 trillion or so microbes per mil of a milliliter of luminal contents within your intestines. So it's an extremely large and diverse population of microbes. So so that's actually been the challenge. So, but what has been wonderful is we now have the ability to sequence the microbes, the DNA from the microbes that are present within your intestine, and we have developed really, I think, really nice tools to describe the complexity and types of microbiota that one individual has versus another. So, and what we found is that the diversity of the microbiota is extremely important. Patients with inflammatory bowel disease, instead of having 1,000 or so species, will have far fewer species. So the analogy that's often used is the rainforest, which has a large and diverse set of life in it, and that if that gets pared back, basically you lose that diversity. So that diversity is actually a strength. There are also, while we focus, I think, a lot on bacteria, that's the predominant microbe that lives in our gut. There are also other types of microbes that are in our intestines that are now gaining increased interest, and these include viruses and fungi as well. And while these aren't as far along, I think that these are potentially going to be very interesting to study and try and decide if they play a role in inflammatory bowel disease. Next slide. Okay, so the initiative, which has been going on for some time, is basically to understand, the simple goal is to understand the role of gut microbes that include bacteria, these viruses and fungi, that play a role in normal digestive health, as well as potentially inflammatory bowel disease. So you have to remember that while there may be microbes that are not beneficial that exist in patients with inflammatory bowel disease, the microbes we normally have that inhabit our intestines are actually quite beneficial to us and provide a lot of benefit. most of which is basically they're able to digest complex carbohydrates that we can't digest. So they allow us to utilize certain food sources we normally couldn't utilize, just as an example. Okay, next slide. Okay, so there has been multiple phases to this microbiome initiative over time. So the first phase is, so you can go to the next slide, has been basically, it basically started out at a very kind of ground level, basically. So, so basically this was developing the techniques to allow scientists to even study the gut microbiome. This is basically the techniques of sequencing specific genes within gut microbes. So this was very important. And then basically be able to take all of this complex sequence information and then basically put it into a form that is more simplified, so that you can compare, say, the microbiome of one individual to another individual, or someone with inflammatory bowel disease without inflammatory bowel disease. So that's really what phase one was basically tool development. And this was very important because I think this has helped not only the IBD field, but it's helped a number of other fields that are interested in the gut microbiome. So the second phase was beginning to decipher the beginnings of what are some of the microbes that are present in patients with inflammatory bowel disease. So again, basically taking broad surveys of microbes that were present, and beginning to understand that this concept then that there were microbes that were clearly beneficial in healthy people that were lost in patients with inflammatory bowel disease, and that there were certain microbes that seemed that weren't typically present in healthy patients that were now present within patients with IBD. Okay, the third phase then was basically to push this further, was to take this beyond simply surveying just bacteria and beginning to expand this out to thinking about viruses and even bacterial products and bacterial genes that were present and what they actually might be, making within the gut lumen that would either be beneficial for healthy people or potentially detrimental for people with inflammatory bowel disease. And then the fourth phase has been advances in knowing the impact of these bacterial products on gut mucosal health. So we're now into the fifth phase of this initiative, which is the next slide, which I think is a pretty exciting phase. So now we've established in the first four phases that patients with inflammatory bowel disease have altered gut microbes. You've lost some healthy microbes, and then you've gained some potentially detrimental microbes that could be causing problems. So it's challenging to actually study a lot of these microbes. Many of these microbes actually can't live within environments that include oxygen. So it's difficult to study the actual microbes themselves. But what we can do is we can actually study the bacterial products that are actually made. And these bacterial products are called metabolites. So we can now broadly survey within the guts of patients with IBD and patients without IBD, and begin to understand broadly what are these metabolites that are enriched in patients with IBD versus healthy people, and begin to get an idea of what processes within the gut microbes then are say overactive, that could be then potentially targets for eventual therapy, and eventually, and hopefully even using this information to predict responses to treatment and development therapies. So the potential here is basically to develop more specific treatments for the gut microbiome to basically help manage IBD, and as it says here, give control back to patients and parents to manage the disease. We think the gut microbiome is a really important kind of output of this particular disease, and if this is something where we can actually develop therapies that specifically target elements of this, this is something that could be, I think, really exciting and really new. Next slide. Okay, so the last part of this is is we're going to talk about ways that you can participate in IBD research.
Thank you very much, Dr. Stappenbach, for that really informative presentation. We really appreciate it. So some ways you can participate in IBD research. Participating in IBD research is one of the best ways to be proactive in helping find safe, effective, and new treatments and give doctors, patients, and researchers a greater understanding of what causes these diseases. I'd like to share some ways you can be involved with IBD research. Clinical trials are organized medical research studies that look for new ways to prevent, detect, or treat disease. By participating in a medical study or a clinical trial, you can have a more active role in your own health care, gain access to new research treatments before they become widely available, and help others by contributing to a medical research. To learn more about clinical trials, visit any of the websites listed on the right-hand side of the screen. CCFA Partners is another great way to be involved in internet-based research. It is a web-based ongoing partnership of over 10,000 IBD patients, both pediatric and adults, aiming at improving quality of life through research and education. With more interactive updates to be featured in 2015, patients will be able to view results on key factors in IBD. Visit www.ccfapartners.org for more information. Now moving on to the question and answers portion of our webinar. Before doing so, we understand that some of you may have had trouble with your audio in the beginning of the presentation, so I just wanted to take a moment to reintroduce Dr. Stappenbach and his background. So Dr. Stappenbach received his MD and PhD from Northwestern University Medical School. He trained in anatomic pathology at Washington University School of Medicine and is board certified in this specialty. He studied host microbial interactions in the intestine as part of his postdoctoral fellow. He serves on the Scientific Advisory Council and as co-chair of the Senior Research Award Study Section for the Crohn's and Colitis Foundation of America. Dr. Stappenbach's research program has focused on understanding the pathogenesis of inflammatory bowel disease. And we thank him so much for participating in today's webinar. We have a bunch of questions that came in. So starting off with one that came in, and just some housekeeping. For everyone's benefit, please try to keep your questions general without many personal details so we can provide an answer that is general in nature. And in the interest of time, I also ask that you keep your questions related to the topic. You are always welcome to contact our IBD Help Center if you have any other questions, and we'll review the Help Center's contact information at the end of the program. So if you have questions, please type them into the chat box on your screen. So Dr. Stappenbach, one question, the first one that came in, it's asking in relation to genetics. So what does current research say about chances of developing IBD if a family member has IBD? What are the chances of another family member developing IBD?
Yeah, that's a really great question. So there have been this has been, I think, an interest of the research community now for decades, basically. And I think some of the very seminal studies were done in the 70s and 80s, basically looking at twin studies. So you can look at twins that are essentially genetically identical, and then basically watch them over time. And then if one of them, so it's basically you start with one of the twins has inflammatory bowel disease. Does the other twin ever develop inflammatory bowel disease? So so how much concordance is there even with the genetics? So looking at patients, so genetically identical twins. So with Crohn's disease, the interesting thing is there's about a 50% chance of getting Crohn's disease. So I think this really shows that the genetics are important, but the genetics aren't everything. That there are still going to be exposures to the environment and triggers that are going to be very important. So that's one piece of data for Crohn's disease. Interestingly, for ulcerative colitis, the twin studies show a lot less concordance. So it's much more on the order of 8 to 10% chance. So if one genetically identical twin has IBD, there's an 8 to 10% chance that the other genetically identical twin will develop the disease over time. Again, so it really suggests that ulcerative colitis has even, the genetics are important, but the environment is going to be very, very important. So then the chances then, so if you're not genetically identical to someone with IBD, say even just a family member, then the chances are even lower than that. So less than, on the order of 5% or less.
Thank you. Okay. Next question. So this is in regards to gut bacteria, and someone's asking, is there any research in finding out if we can put back the good gut bacteria that is prevalent during remission and missing during flares as a way to control flare-up? So I guess speaking to kind of the gut bacteria balance.
Yeah, I think this is something that's very much on people's minds, is how can you, because the studies basically surveying gut microbes have shown this imbalance in the microbiome. Can we simply add back the good bacteria? And one method that's basically generated a lot of buzz and interest has been fecal transplantation. So basically, what this involves doing is basically making a slurry of feces from a donor, and then basically infusing this into the intestine of someone with a disease. So where this has been very successfully used has been in patients with Clostridium difficile. This is something that's actually worked quite well at kind of basically pushing out a pathogen that can cause disease. Whether or not this will work in inflammatory bowel disease I think is an area of intensive investigation. But I think it's something that's definitely many people are actually thinking about. So that would be one way to do it, would be to simply add back, say, donor feces from someone who's healthy. I think maybe a more sophisticated way of thinking about this is if we could actually identify and grow in the lab the bugs that we know that are beneficial. We might be able to make something that doesn't require a donation from another person, and that we could make it be I think a little more specific and scientific. So I think that's another potential way to think about this. The trick is, though, is it's possible that this type of approach may not be durable. There may be effects of the genetic mutations in your intestine that will make it hard for these bacteria to recolonize. So this is something that I think this is where a lot of this work will go. I think over the next 5 to 10 years, where we can basically begin to try and understand exactly how to get to reestablish essentially what is called homeostasis, where you basically have normal gut microbes that are then in close contact with the inner lining of your wall, and reestablishing that good community. And how to do that, I think, is going to be what's going to be really exciting and interesting.
Thank you. And kind of piggybacking off that, I know you mentioned in the presentation importance of diversity in the microbiome. So someone had asked, can probiotics help with the diversity in the microbiome?
That's a really great question. So probiotics typically these are lactobacilli. These are things that can either be taken in pill form or they can be placed into various foods. And they've been shown in animal studies to have a variety of benefits under conditions of injury, such as if you take a mouse and you irradiate it, these probiotics can provide some benefit to repair. It's not really clear, though, yet how these probiotics actually act. So I think that's something that's a little bit of a mystery. The other thing is that the probiotics that you're given, these are essentially microbes that don't stably colonize your intestine. So you would take a pill that would have these probiotics in them, and basically, essentially, they just pass through your intestines. So while it's possible they may have some benefits by the metabolites that they make, this is, again, something that will not be stable, because these microbes, again, can't stably colonize your intestine. The microbes I think that we're most interested in right now are populations of microbes that basically can live in and around the mucus that lines the lining of your gut, and trying to figure out how these microbes actually live there, and then how to replenish. These are the microbes that are lost, and these are the microbes we would basically like to replenish in patients with IBD. So that the probiotics that you can currently buy won't do this, and they really won't increase the diversity of your gut microbiome.
Okay, and next question. Do you think that the antibiotics can play a part in killing off the good bacteria that can cause this kind of unbalance in the gut?
Yeah, so antibiotics, I think, so when you think of having a bad bacteria, you think of an infection, right? And so something where you'd find a bacteria or an antibiotic that could kill that particular pathogen, something that was causing, say you had bacterial pneumonia or something like that. You'd want to give an antibiotic that would kill the pathogen. Here, it's a lot. It's not so simple. It's basically not a single organism that appears to be acting like a pathogen that's causing the problem. It's basically an imbalance of microbes. So you have a wide array of microbes that normally live in the environment that are now living within your intestine. And the question is can you use antibiotics then to basically try and wipe those microbes out? And that's actually been pretty tricky to do. It's I think the microbes that don't belong in your gut, it's very difficult to find single antibiotics that could target them and basically wipe them out without doing collateral damage to the existing good bacteria that are still present within your intestine. So I think while I think antibiotics are used with patients with inflammatory bowel disease, I think it's been very difficult to basically to look at their effects in a systematic way. So this has been, I think, really problematic. And I think this is really one of the goals of the microbiome is to find really more specific metabolites and bacterial products that could be targeted by specific therapies. And this might give us a way to more specifically go after what the bad bugs are actually making instead of actually giving broad-spectrum antibiotics. The other problem is the other question is can antibiotics be potentially leading to some of the problems that we're actually seeing? So say the indiscriminate use of antibiotics and particularly the pediatric population. And there's some really interesting work from Marty Blazer's lab looking at the effects of antibiotics in early life. And the idea is by giving someone who's healthy an antibiotic, it basically alters your gut microbiome, and it does so. The effects of altering your microbiome basically are pretty durable, surprisingly durable. So it's not when you stop taking the antibiotic, your gut microbiome goes back to normal really quickly. It can actually be altered for quite some time, and then the idea is with repeated antibiotics, this is something that could be become even more severe. So I think on the flip side, thinking about antibiotics is they actually may be causing or be one of the factors that could be actually basically helping predispose people to this disease through use in early life. It's a really interesting idea.
Thank you. Okay. All right. Next question. We have a question from a patient with IBD who's a little bit older, and she's wondering, can age affect, or is there any research that it talks about age and IBD and how the microbiome or genetics may differ in older patients that develop IBD?
Yeah, that is a really, really important question. I mean, because I think some of the most interesting patients are patients that develop the disease very late in life. There's definitely a spike in IBD that occurs after 50, and so the question is how can you live your whole life and be healthy and then develop this disease, and what is it about the older individual then that can help predispose to this? And I think this is something that has not been carefully examined. I don't have a good answer for this particular idea, but it's something that I've thought about, but have not actively pursued. But it's a really interesting idea. I think a lot of people are very interested in the fact that a lot of the where inflammatory bowel disease is really increasing in incidence has been in kids, and particularly young kids, where we didn't see that many cases of this, say, 10, 20 years ago. Now it's becoming much more common. So I think that's where a lot of the attention is turned, trying to figure out why young people now develop it so much more frequently. But the flip side is trying to understand why older people develop it. I think is really interesting.
Yes, okay, thank you. Next question, are there any common epigenetic changes that are seen in both individuals with Crohn's disease and ulcerative colitis?
Wow, that's a great question. So epigenetic changes are going to be basically these are modifications to your genome that all occur basically very often in response to elements of the environment. So these are essentially modifications of your genome, things that you're not born with that then become imprinted on you and on your genome. This is something that my lab is particularly really interested in. We're particularly interested in this epigenetic changes and the cells that line the wall of your gut, the intestinal epithelial cells. So the lining of the intestinal epithelium basically is driven by intestinal epithelial stem cells. These cells live basically sequestered away from the lumen of the gut and things called crypts. So these stem cells then divide and produce daughter cells that then differentiate and then form a barrier that's between your gut microbiome essentially and you, the rest of you. So this epithelium is clearly abnormal in patients with inflammatory bowel disease. We've known this for decades, and so the question is, is this abnormality something that is constantly being driven by external factors, or did the stem cells essentially become imprinted with these epigenetic changes, and then basically they then will always continue to produce abnormal epithelial cells? It's going to be really important to understand this distinction, because if there are epigenetic changes, the types of therapies that we might actually develop to try and correct the epithelial abnormality that's present in patients with IBD would be very different than if it wasn't imprinted with epigenetic changes. If it's not imprinted, we'll very likely try and figure out there must be some consistent cause that's stimulating this abnormality, so we need to figure out what the environmental stimulant is. If it is epigenetic, then we can begin thinking about basically new types of therapy to actually attack these modifications to the genome and try and fix this. So my lab is actually testing this idea out now, and we should hopefully have a pretty good answer on this within the next 6 months to a year. And so stay tuned.
Okay, great. We have time for a few more questions. So the next question is asking in relation to research on diet and the microbiome. So is there any research or any findings that indicate that a certain diet or certain foods may impact the microbiome?
Yeah, so I think the best authority on this is, or one of the best authorities is Gary Wu at the University of Pennsylvania, and he always points out. I've heard him talk about this topic this many times, and he's definitely an expert in this area. And what he points out is that we know that we can improve symptoms of inflammatory bowel disease by using something called an elemental diet. A very simplified form of nutrition. It's very bland. It tastes awful, but it's something that can basically improve IBD. So in principle, you can affect IBD with diet. The tricky part, though, is how to actually put this into practice. beyond using things like elemental diets. And I think that that's where this has been very, very challenging. And I think the goal of Gary's work and others' work is to try and actually figure out, are there components of the diet that actually can say affect the microbiome, which can in turn drive or basically perpetuate inflammatory bowel disease. But I think in principle this is something that can happen, but I think the details of it I think are still beginning to emerge. And this is actually, Gary's part of the Microbiome Consortium with the CCFA, and he's been a wonderful colleague to have in this particular area.
Thank you. Another patient has asked, can you give some examples of metabolites that predict disease activity and response to treatment?
Yeah, that's the million-dollar question. So that's I would love to have that answer for you today. So what we have now are lists of metabolites that we know that are enriched in patients with IBD versus are predicted to be enriched in patients with IBD versus metabolites that may be diminished in patients with IBD. And what we're currently doing now is we're currently functionally testing these metabolites in relevant cell types in vitro. So my lab is really good at growing human intestinal epithelial stem cells and then differentiating them on a variety of platforms. So we're testing the effects of these potentially IBD-enriched metabolites on our epithelial cultures to see if they can do anything to the epithelial cells that makes them abnormal. There are other colleagues of mine within the consortium that are testing these on inflammatory cells, again, to test the function of these metabolites. So the goal is over the next 6 months to a year is to basically get exactly this list, exactly the answer to this question. And we hope to have a pretty good idea of which metabolites will be beneficial and which will be really harmful to really critical cells within the gut, and then begin designing additional tests and maybe even thinking about specific targets for these metabolites. But that's a great question.
Thank you. Okay, next question. Do you foresee genetic testing becoming available to see if a person is at a higher risk of developing IBD or if they are at a higher risk of developing complications from their IBD?
Yeah, this is another really great question. So I think so the interesting thing about the susceptibility genes, most of these genes actually only give you slightly higher risk. So a classic gene in this particular space is an autophagy gene called ATG16L1. And if you have mutations that are risk for this particular gene, your chances of getting IBD are only maybe 30% higher than if you didn't have the gene. So the question that I think geneticists are really interested in is basically, can we then, since individual genes, since they seem to have relatively small effects, is there something about the kind of the global effect of multiple risk alleles? So I know colleagues of mine, such as Dermot McGovern at Cedars-Sinai, he's a geneticist and he's really interested in this question. So he's wondering if you can develop essentially a risk score that will then sum all of the potential risk alleles that a person will have, and then essentially come up with some sort of score that you can then know you have. So I think, yes, I think this is something that could happen. We're still a little ways off from it, but I think you know, the ability to really finely genotype people is really coming. And even sequencing people's genomes is really coming. So I think we'll have this information. I think the answer is yes.
Thank you, Dr. Steppenbach. Another question that's come in relation to genetics says, can a gene that may play a role in getting inflammatory bowel disease be turned off?
Right. So if you have the gene, can you actually say do gene therapy? Is that a possibility, or could you find a drug that could actually alter it? So there are two types of scenarios that we've seen with the mutations that are associated with IBD. In most cases, these mutations seem to be associated with less function of a gene. So these are cases where we would like to basically find drugs that promote the function that's missing when this gene doesn't function. So in one case, we're looking for drugs that can basically stimulate. And the genes, but there are examples actually where genes actually they function by their mutation basically turns them on too much. And in this case, what we actually want to do then is to try and find drugs that actually kind of dampen the activity of these particular genes. So I think the way to think about this is basically a lot of this will become fine-tuning. It's not that the gene is necessarily on or off, but it's that there's too much or not enough activity. So the way we're thinking about this is to basically design drugs that can either promote or inhibit activity based on the types of mutations that are present.
Thank you. Okay, we have time for probably 2 or 3 more questions. One other question that's come in, it's asking, do you anticipate the use of stem cells in the treatment of IBD, or are you aware of any research on stem cells in IBD?
Oh, that's a great question. So I ran a workshop on stem cells at the recent CCFA sponsored research meeting, and there was a tremendous amount of interest in stem cells. So there are a variety of stem cells in your body. So there's a couple of ways to think about this. There are stem cells in your body that basically generate certain cells that populate your organs. So you have stem cells in your bone marrow that make all of your blood cells. You have something called mesenchymal stem cells that are present in a lot of your tissues, and these are very interesting stem cells. And then there are things like epithelial stem cells that regenerate the linings of organs, like the intestinal epithelial stem cells. So that's kind of one group of stem cells that people are very interested in. And another group of stem cells that people have become very interested in are induced pluripotent stem cells. So these are stem cells basically what you can do is take a fibroblast from someone, and then you can figure out ways to re-engineer it so that you can make it turn into whatever cell type you would like it to turn it into. This type of technology is still in its infancy, and the ability to re-engineer these cells, say to take a fibroblast and make it into a gut epithelial lining cell, is still in its infancy, but there have been some really interesting breakthroughs in using these particular cells. So the goal then, I think there are then two, so once you have these stem cells, I think there's a couple of interesting goals that could actually be, where these cells could actually be utilized. One is that you could actually begin to think about repairing really heavily damaged areas of tissue. So could you actually basically engineer the intestine, say, in vitro, and basically replace parts of the intestine that have been so damaged that they need to be removed? So that's something that's a possibility. I think down the road, I think that actually could happen. The other thing is that the other more immediate use of stem cells, I think, is the mesenchymal stem cells. These I think have been very intriguing. These are cells that can basically be injected on the bloodstream. They then basically can make anti-inflammatory molecules, and they've been very successful in treating a number of inflammatory conditions. The problem has been trying to treat inflammatory bowel disease with mesenchymal stem cells thus far has failed. And I think from my perspective, the main reason is it's been very difficult to deliver these cells to the precise areas where they need to be delivered. So I think there are a number of labs actually working on thinking about how to deliver these cells more precisely to areas of damage. And I think I personally think that they offer a really kind of wonderful opportunity to deliver cellular therapy for inflammatory bowel disease. The other part of stem cells could be diagnostic. So the ability to grow intestinal epithelial stem cells from patients is now something that we can do. I have in my own lab, I now have over 200 lines of stem cells from patients with and without inflammatory bowel disease, and we can essentially use these as platforms, we think, to basically begin to try and stratify patients based on response to particular cytokines or bacterial products in the lab. So this is something that we're very excited about. So we think that if they, in some cases there may be some therapeutic use for these, but there may also be some diagnostic and prognostic uses for these stem cells as well. So it's a very exciting time for looking at stem cells within this particular disease.
Thank you so much, Dr. Stappenbeck. So this is going to be our final question of the evening, and it's kind of a nice way to wrap up. Someone has asked, how can the research that you've talked about today on the microbiome and the genetics initiative translate into treatment over the next 10 years?
Right. I think that's really the whole goal, is to basically be able to do this. I think the simple short answer is I think the specifics now that we know behind the genetics and the specifics that we're learning about in the alterations in the microbiome are going to give us new targets for therapy. This has really been the wonderful and major breakthrough that's happened over the last decade. Is we've had this. We knew that genetics played a role. We knew the microbiome played a role, but we didn't know the specifics of how this actually might happen. And now what I have, what I feel like as a researcher, what I have open before me is I have all of the potential characters in front of me. And now all I need to do is figure out for specific genes and for specific alterations in the microbiome, how can we use this information then to go forward and develop new therapies. So I guess the short answer is now we have targets, and now we just need to figure out how they function, and then how to actually target them in the proper cell types. And that, I think, that's the promise of these initiatives.
Thank you so much, Dr. Stappenbeck, for your insightful presentation and answer to all of these patient questions. We got through so many today. Anyone's question that wasn't answered, you are always welcome to contact the IBD Help Center if you have additional questions. So in closing, we again would like to thank Dr. Stappenbeck for his answers and his presentation on the matter. To learn more about other CCFA research initiatives or ways you can support IBD research, visit the website listed below. The Crohn's and Colitis Foundation of America is also available to offer education and support. The IBD Help Center is open Monday to Friday, 9 a.m. to 5 p.m. Eastern Time by calling 888-694-8872 or by emailing info@ccfa.org. You can also chat online with an information specialist directly via Answer Chat by visiting www.ccfa.org. If you'd like to watch other educational webcasts on IBD, please visit the websites on the screen to explore other IBD-related topics. You can also connect with other IBD patients and engage in discussion through the CCFA community website, support groups, and Power of Two peer-to-peer mentor program. Another resource is our GI Buddy. It's a tracking tool and a mobile app that has everything you need to stay on top of managing your inflammatory bowel disease. Visit www.ccfa.org for more information. Finally, participate in other education events by connecting with your local Crohn's and Colitis chapter. Visit the CCFA website for more information. Please remember, an archive of today's program will be posted on CCFA's website within the next month. Upon exiting the webcast platform, you will be prompted to complete a post-program evaluation. We ask that you please take a few moments to provide your responses, as your feedback is extremely important, and it helps us to plan for future educational activities. On behalf of the Crohn's and Colitis Foundation of America, we thank you all for joining us tonight. Goodbye.